Hello everyone, today we're continuing our discussion of the development of tunicates, which is a type of deuterostome invertebrate, and we'll also be discussing the development of amphibians, which are vertebrates. Now recall from last class that we talked about the fact that tunicates or sea squirts are invertebrate chordates, which means that during development and also as a free-swimming larva, before metamorphosis, tunicates have a notochord, which degenerates when the organism undergoes metamorphosis. However, since these animals lack vertebra, they are not considered vertebrates even though they possess a notochord during some stages of development. Like sea urchin, tunicate embryos also exhibit complete or holoblastic cleavage. However, the cleavage pattern of tunicates is bilateral, which means that the first cleavage actually cuts or bisects the zygote into the left and right halves. The following cleavage planes are centered on this axis and result in the two halves being mirror images of each other. The beginning at the second cleavage, you see that the cells are divided asymmetrically so that the anterior cells are larger than the posterior cells. These asymmetric cell divisions continue until the embryo has 64 cells. So at each cell division, the posterior blastomeres are always going to be smaller compared to the anterior ones. These asymmetric cell divisions are made possible by the presence of a structure that's known as the centrosome attracting body, or CAB. The CAB is composed of a mass of cortical endoplasmic reticulum, which is what you see in this image labeled in red, and mRNAs. And in this image, it is shown as a CER mRNA domain. As you can see from this figure, during mitosis, particularly during metaphase, the CAB becomes associated with the posterior centrosome. And it also connects to the membrane via PAR proteins, which are depicted here in purple. Now that leads to the pooling of that posterior centrosome near the membrane so that the posterior cells end up being smaller than the anterior cells after cell division. Another function of the centrosome attracting body is to attract mRNA so that they will end up in the posterior most cells at each division. So the CAB serves two roles here: uneven spindle positioning so that you end up with cells of different sizes, and also the segregation of localized determinants so that each daughter cell has a different fate according to the mRNAs it inherits. In the tunicate, most of the early blastomeres are specified autonomously, which means that different regions of the early embryo are going to inherit specific cytoplasmic determinants in the form of mRNAs. Now, before fertilization, you have a yellow cytoplasm that's located in the cortex of the oocyte, and that surrounds a gray cytoplasm in the interior of the oocyte. After the sperm enter the eggs, which only happens in the vegetal pole, meiosis resumes, and when the nuclear envelope breaks down, there is a clear cytoplasm that is released and it displays to the animal pole. And within a few minutes of sperm entry, the inner and the cortical cytoplasm have actually moved to the vegetal pole. Then, as the male pronucleus migrates to the equator to meet with the female pronucleus, the yellow cytoplasm migrates along with it, and that's made possible by the reorganization of microtubules, which happens, or it's done by the sperm's centrosome. And so you end up forming this yellow crescent here, which extends from the vegetal pole to the equator. This yellow crescent is the region that contains the mRNA for the transcription factor that's required for the specification of the tail muscles of the tunicate larva. And the cells that receive this yellow cytoplasm are going to give rise to most, but not all, of the mesodermal tissues. Now, the first cleavage that cuts these two cells, or the oocyte, into two halves is going to also divide the cytoplasm, the yellow crescent cytoplasm, among the two sides. Subsequent cleavage causes the cells that will give rise to the mesoderm to be located in the posterior portion of the embryo, while the cells that will give rise to the notochord and the nervous system are localized to the anterior. As we have seen before, both conditional and autonomous specification are observed in the tunicate. While the gut endoderm, the muscle mesoderm, and the skin ectoderm are autonomously specified, the nervous system, the notochord, and the heart and mesenchyme are conditionally specified. This figure depicts that in part, since separating the cells of the tunicate embryo at the 8th cell stage, which is what you see here, leads to the formation of most tissue types but not the nervous system. Now notice here that even though the notochord is conditionally specified, it still appears to form properly when the cells are separated early in development. However, the reason for this is that the signals that induce the formation of the notochord come from the endodermal cells, which are going to be derived from the same blastomere that gave rise to the notochord, as you can see here. On the other hand, since the nervous system comes from a blastomere that does not also give rise to the notochord, the cells that would normally be induced to form the nervous system from signals that are going to come from the notochord are not going to be exposed to those signals and end up forming skin ectoderm instead, which is the default fate of the ectoderm in tunicates. Now let's look more closely at some examples of both autonomous and conditional specification in the tunicate embryo. As I previously mentioned, the cells that give rise to the tail muscles are autonomously specified, and those cells are derived from the yellow crescent region of the oocyte. They contain an mRNA for the transcription factor MACHA1, which is shown here in part 8. And that transcription factor is required for the activation of muscle-specific genes, including muscle actin, which is what's labeled here in the figures in part B. Now, if you were to transfer MACHA1 containing cytoplasm to blastomeres that do not normally give rise to muscles, as shown in this figure, you would see that these blastomeres would be able to give rise to muscle cells, as evidenced by the presence of muscle actin in those cells. On the other hand, depleting MACHA1 from the blastomeres that normally give rise to muscles makes it so that these blastomeres are no longer going to be able to differentiate into muscle cells. Now, another function of MACHA1, besides turning on the transcription of genes that are required for the specification of muscle cells, like muscle actin, is to inhibit the expression of genes that are required for the formation of the notochord. Now, it appears to do this by acting together with the transcription factor TBX6 in a feed-forward manner. So that means that MACHA1 will turn on the expression of TBX6, and that then MACHA1 and TBX6 together will turn on the expression or activate the expression of another transcription factor, SNAIL. And this transcription factor SNAIL then acts to promote muscle fate by activating other transcription factors that are required for muscle specification, and it also acts to inhibit notochord fate, specifically by inhibiting the transcription factor brachyury, which is required for notochord specification. So the effect of this is to prevent the muscle precursors from becoming notochord cells. The endoderm, like the muscle cells, is also autonomously specified, and it's derived from the vegetal blastomeres. Now, the specification of the endoderm requires the presence of the transcription factor beta-catenin, just like in the sea urchin. Now, this figure shows the lack of beta-catenin in the nuclei of the animal pulse cells and the presence of beta-catenin in the nuclei of the vegetal pulse cells. As in the sea urchin, inhibiting beta-catenin will result in the loss of endodermal structures that are going to become replaced by ectoderm. On the other hand, if you were to overexpress beta-catenin, as we saw in the sea urchin, that would lead to the expansion of the endoderm at the expense of the ectoderm. Finally, when beta-catenin is expressed in notochord precursor cells, which are labeled here by the black arrows, those cells will become endoderm and express those endodermal enzymes, which is the dark staining that you see here. The white arrows, on the other hand, indicate the normal expression of the endodermal enzymes in the tissues that will typically give rise to the endoderm. So we've looked at the autonomous specification of the endoderm and of the tail muscles, but some of the tissues of the tunicate are conditionally specified, and two of these tissues include the notochord and also the mesenchyme, and both of these are mesodermal tissues. Now, the induction of both the mesenchyme and the notochordal fates requires the paracrine factors of the fibroblast growth factor family or FGFs, and those FGFs are going to be secreted by the endoderm. Now, beta-catenin expression in the endoderm is what's indirectly responsible for the secretion of the FGFs. It activates the second transcription factor that then leads to the secretion of these FGFs, and the FGFs in turn induce the expression of brachyury, which is a gene that's important for the specification of the notochord. Now, the cells that are going to give rise to the notochord and also the cells that will give rise to the mesenchyme are both going to be the descendants of cells that are going to be located near the endoderm. So the endodermal cells will be adjacent to the cells that are supposed to give rise to the notochord, and they will also be near the cells that are supposed to give rise to the mesenchyme. So both the notochord precursors and also the mesenchyme precursors are receiving FGFs that are coming from the endoderm. So what is it then that allows some cells to differentiate into notochords and other cells to differentiate into mesenchyme, even though both cell types are receiving FGFs from the endoderm? What determines whether the cells that are going to be exposed from FGFs coming from the endoderm are going to be mesenchyme or notochord is the presence or the absence of the MACHA1 transcription factor. And the vegetal pole, where the cells receive the MACHA1 transcription factor, those that also are exposed to FGFs coming from the endoderm go on to give rise to mesenchyme. Those cells that are located farther away from the endoderm, so they have the MACHA1 transcription factor, but do not receive the FGFs coming from the endoderm, go on to give rise to muscle cells. On the other hand, in the anterior, where MACHA1 is absent, the cells that receive FGF from the endoderm go on to develop into the notochord, while those cells that are localized even more anteriorly and those are too far from the source of FGF and do not inherit MACHA1 go on to give rise to the nervous system. Finally, although it's not shown in this figure, there are other factors that are required for the induction of the nervous system that are going to come from the cells that give rise to the notochord.